Okay, Austin said he will be in the lab Monday through Thursday at 7 until whatever, proctoring. So if you need to come in and do any work, come in. Okay, big push last week. Okay, today is going to kind of put together everything we've been building up to for the last couple of weeks. What I want to do now is kind of talk about how we get the interaction between all of these cells and cytokines and receptors necessary to carry out our adaptive immune response. Remember, the adaptive immune response is one, we're born with elements of it, but it doesn't reach full fruition until your body has gone through an education process, your immune cells have gone through various developmental sequences, and this is all really the result of being challenged by particular antigens. And so this is the adaptive immune system. Excuse me. We talked last time about the fact that there are two overlapping arms or segments, and the fact that they're overlapping is really very important. Cell-mediated immunity is involved with T-cell interactions. Additionally, antigen-presenting cells are involved in this. On this particular slide, when I talk about the kind of cells involved, I'm really only referring to the lymphocytes that are involved. But there are antigen-presenting cells involved in cell-mediated immunity as well. This is in response to pathogens that will infect cells or cells that become cancerous. So this is a way for the immune system to kill your cells that have become damaged. Humoral immunity is B and T cell mediated. The B cells are the ones that are actually responsible for producing the antibody, but typically they need assistance in the form of a T helper cell in order to be able to do that. We'll see, though, that there are exceptions to that particular rule. So let's take a look at cell-mediated immunity first. What's happening in cell-mediated immunity? Well, as I said, we're killing damaged host cells. And the major player here in terms of carrying out the killing are the T killer cells or T cytotoxic cells. Same thing. Okay? And in fact, I think in a lot of my abbreviations, sometimes I'll do this, sometimes I'll do this. Okay? Synonymous terms. However, T cells are also involved in helping B cells produce antibody. In both of these instances of T cell action, this actually involves a fairly complex cooperation between antigen-presenting cells, maybe it's a macrophage, maybe it's a dendritic cell, various T cells, and various B cells. So we're going to be looking at some slides that kind of shows you the interplay between these. And it's going to get a little complicated. And there are going to be instances where there are things on the slide that are there for illustrative purposes, but not things I expect you to really know for the exam. And so that's why over here on the side, I've kind of done a big picture summary of what some of the really most important parts are. And so you definitely should get this down, because this is a way you can start making sense of the complexity of what we're going to talk about. Kind of boiled down to some stuff you really do need to know for the exam. Okay, so make sure you get that. And we'll be referring to that kind of as we go along. Okay, let's take a look at some of the players in cell-mediated immunity. Here's our antigen-presenting cell that we're going to talk about most commonly, and that is the dendritic cell. The dendritic cell, remember, is a cousin, if you like, to the macrophage. And these particular antigen-presenting cells have two important functions. One is to phagocytize the antigen. And in doing so, they will end up presenting it on the surface of their cells. But they can only present it in the context of an MH2 receptor, an MHC2 receptor. The other major function of these particular cells is to produce cytokines. And those cytokines will have an effect on other immune cells. So I've got a couple of the important cytokines that are produced by various players in this process. So for example, interleukin-2 is one of the cytokines that's produced by the antigen-presenting cell. Okay, there are various types of T cells. And this really only shows 3 of the 4 types. Up until probably the last 3 or 4 years, this would have all been lumped together just as T helper cells. But one of the things that we're finding right now is that there are actually subsets of T helper cells that play a little bit different role in different parts of the immune system. So we'll dig into that just a little bit. Both of them have a CD4 receptor. Okay, we've got some important receptors down here. And CD4 receptors are found on T cells, down there. That's better. CD4 cells are found on T helper 1 and T helper 2 cells, and CD8 receptors are found on the T cytotoxic cells. It's important for you to understand that these CD4 receptors on the T cell interact with an MHC2 on an antigen-presenting cell. So this is a way that those two cells find one another. CD8s associate with MHC1, CD4s associate with MHC2. So this is the basis of MHC restriction. That's something we talked about when we talked about MHC receptors. T cytotoxic T killer cells are MHC1 restricted. T helper cells are MH2 restricted. Over here, you'll see some of the major functions or important features of those various T cells. And down here, you can see that the T cytotoxic cell is actually the one that does the destruction of the damaged cell. These particular cells, the helper cells, influence other immune cells. So T helper 1s, for example, can secrete some interleukins. Interleukin 2 here, for example, is shown. And they can be responsible for activating certain other types of cells. T helper cells 2 activate B lymphocytes, and they produce their own battery of interleukins. But again, there's more information over here than you probably need to know. I'm really concentrating on 3 cytokines: interleukin 4, interleukin 2, and interleukin 12, which isn't shown right here. And these are the major cells that produce those particular types of cytokines. So sometimes it's a little bit confusing, and I run the risk of providing you too much information in some of these slides. So please understand that we're trying to narrow it down and boil it down to some basic concepts, which are illustrated over here. Okay, what are the general steps that are involved in activation of the T killer cells? Well, we've got to show an antigen to some cells to get the T killer cells activated. That antigen is shown by an antigen-presenting cell, a dendritic cell or an macrophage. Presenting that antigen to those cells turns them from inactive cells to active cells. Now remember what being inactive involves. You're educated. You have the receptor, but you're in a kind of state of dormancy. And until you're presented that particular antigen, that cell won't then start to divide and go through that clonal expansion process. So once you're activated, you go through clonal expansion. And then, this is kind of a minor point, but these cells, once they become activated and dividing, actually start self-stimulating themselves. And the cells produce substances that enhance the process. So this is really a positive feedback phenomenon that's occurring right here. And that helps to amplify and really ramp up the immune system. Okay, so here's one that's going to look a little bit complicated. Unfortunately, it gets worse. Okay, some of the other slides. So let's start looking through it. Okay, this DC is a dendritic cell. That dendritic cell has both MHC2 and MHC1 receptors on its surface. Okay, remember, all cells except red blood cells have MHC1, but only certain professional cells like the dendritic cells, the macrophages, and the B lymphocytes have these MHC2s. These guys have encountered an antigen, have internalized it, and displayed it. Okay? And in this particular case, the antigen is being displayed both on the MHC1 receptor and the MHC2. Once it's done this, it migrates and will go someplace in the lymphatic system. It might have encountered the antigen in the skin, for example, but now it migrates. And let's say it ends up in a lymph node, and sitting in the lymph node are these inactive T helper cells. When they're inactive, they're usually referred to as a T helper knot. Okay? And they might encounter also some inactive T cytotoxic cells. And remember, these have their receptors, but they haven't been stimulated into action. So in this particular case, this dendritic cell is encountering simultaneously both an inactive T helper cell and an inactive T cytotoxic cell. And it shows those antigens that it's engulfed and is now displaying to those two cells simultaneously. This one in the context of an MHC2, and this one in the context of an MHC1. And all this is showing you over here for this cell is some of the interaction that's taking place between the receptors. Okay, so here's our T cell receptor down here. It's binding a particular antigen. And then here's its CD8, and that recognizes the MHC1 of the dendritic cell. So everything's matching up and pairing up. Okay? So now this cell has seen its antigen. So it starts getting kicked into activity. And it goes through the process of clonal expansion. These particular cells become activated to start being killers, and some memory cells are being produced simultaneously. However, this doesn't happen just as a result of this binding. This binding over here is also essential in order to get the activation. Okay. This basically provides one signal, this binding right here. A second signal is produced by the production of these cytokines. Where are the cytokines coming from? Okay. This inactive T helper cell, this T helper knot cell, is showing the antigen with the MHC2. That causes the dendritic cell to start producing an interleukin. Okay, when these two receptors bind, it activates the production of interleukin here, interleukin 12. And that causes this T helper knot cell to differentiate into a T helper 1. And the T helper 1 is going to produce this interleukin 12, which is responsible for the second signal to getting us into this clonal expansion and cellular differentiation. Confused? It's the kind of thing you really just have to sit down and look at the diagrams and kind of work your way through, but do it with this by your side. Okay? This is the important stuff that you have to be able to kind of reconcile from that particular slide. Yes?
You said interleukin 12 is produced by the Th1 cell,
Mhm.
right? Why does it look like it's being produced by the dendritic cells in the picture?
This is 12 up here. This
Right.
is 2 down
Yeah, you said 12 is produced by Th1.
here. No, 12 is produced by the dendritic cell. Yep. Oh. You're catching all kinds of mistakes here. Good catch. So this is backwards. So interleukin 2. You know what you're going to find when you start looking at these diagrams? Is that there are inconsistencies in some of the diagrams, and I'll try to point out where those inconsistencies are. They're from different sources, and unfortunately, this stuff about immunology is kind of evolving and is really not completely settled. And so I will constantly find conflicting information. And what I'm basically trying to do is go with the sources that are the most in agreement right here. This particular interleukin-2 is important in the differentiation of these T killer cells. And what's kind of interesting is that under the influence of interleukin-2, these cells actually increase their number of interleukin-2 receptors. Down here, part of the differentiation process is developing a new receptor that will respond to this cytokine. Once you then start expanding, these cells producing interleukin-2 cause more interleukin-2 receptors to develop, meaning they become more responsive to the thing that they're producing. And so this is this positive feedback that I talked about, and this is the self-stimulation that occurs. What about this killing mechanism here? What's going on? What's so special about these particular cells once they've gone through this whole activation process? Activated T killer cells will then recognize damaged host cells that are displaying foreign antigen on their surface with an MHC1. So this CD8 right here is going to bind to the MHC1, and that's going to hook those two cells up. Don't worry about over here, this particular receptor complex. We're not going to worry about that one. So what do these activated cells do? They start producing two proteins, which are really important. One of them are called perforins, and the other are called granzymes. When these two cells hook up, that stimulates exocytosis, and these vesicles containing the perforin release the perforin into the space between the two cells, and that perforin aggregates on the surface of the damaged cell and basically creates a channel. What does that look familiar to that we've seen before? A bunch of proteins aggregating on the surface of a cell and creating a channel. Remember the attack complex and complement cascade? Okay, so it's a similar kind of phenomena. But what happens in this particular case is the channel allows the second protein that's being secreted, this protein called granzymes, and they go through the perforin complex, and they activate enzymes in the cell cytoplasm, which had previously been inactive, that cause apoptosis. So this cell shrivels up and dies. Okay. So this cell right here that's been activated now starts producing these things. When it's inactive, it doesn't. When it recognizes a damaged cell, because that damaged cell is displaying a foreign antigen on its surface, maybe a cancer antigen, maybe a virus antigen, those two receptors recognize one another, and that stimulates the formation of the perforin complex, and then the granzyme that's produced enters through that pore and induces apoptosis of the cells. Here is a cytotoxic T cell attaching to a cancerous cell, and so this is in the process of releasing those particular granzymes into this infected cell, resulting in programmed cell death. Okay. So in this particular part of the defense mechanism, the cells have really already been damaged. And we're just trying to destroy the damaged cells so they don't proliferate and produce more damaged cells. Obviously, that's going to be important in halting the growth of a tumor. That's also going to be important in short-circuiting the life cycle of a virus, because if we can kill these cells before they release new virus particles, that stops the replication cycle. what's needed to make this happen. Antigen-presenting cells have to find the antigen first. They have to show it to these cells to start activating them. But the activation process involves another player, the T helper cell, and it itself has to become activated before it can help in the process. Now you're really confused, right?
How do the memory cells help in like a faster response to something?
Because the memory cells don't have to go through some of this initial chemical stimulation. It's really just kind of a shortcut, if you like, almost. And it takes significantly less amount of time. Yeah.
How come, when it comes to cancerous cells and presenting a cancer antigen, all of that, how come is it so ineffective?
Very good question. So why doesn't the immune system destroy more cancers? Is that your question?
Yeah.
Oh, I think it really does. And in fact, probably it's not uncommon at all for all of us to constantly be developing, because of mutation, damaged cells which begin to develop into tumors. But our immune system destroy those before they really become a problem or become noticeable. So for the most part, cancers are wiped out before they really ever get going. But that's the $64,000 question. It's why do some cancers escape detection by the immune system? And one of the things that they do, we think, is that as they mutate, they start messing around with the receptors on their surface, which interferes with binding of the cytotoxic cells or helper cells to them, or the antigen-presenting cells, and makes it harder for other cells to see. So they're constantly changing their surfaces and trying to escape detection.
But in every cancer, no matter what it is, it'll still present cancerous antigen.
Yep.
Okay, so
Yep.
it's smart, pretty much.
Mm-hmm. It's smart, yep. Okay, humoral immunity. Remember that humoral immunity, the goal is to produce antibody. And the reason that we want to produce antibody is that it helps to remove the antigen. Remember we talked about the fact that it can neutralize virus, it can agglutinate cells, it can stimulate the complement reaction. There's a lot of different reasons why production of antibody is beneficial. Well, this results in a cooperation between macrophages, dendritic cells, T helper cells, and B cells. So this is a pretty complicated set of interactions as well. You ready? Okay, we talked about the fact that there are two types of antigens. There are T-independent antigens, and there are T-dependent antigens. T-independent antigens are really simple. You'll like this one, okay? Because they stimulate B cells without T helper cell involvement. They can directly cause an inactivated B lymphocyte to start producing antibody. These particular molecules are typically ones with repeating subunits. They're very simple in terms of their molecular complexity. When they bind to the surface of the cell, one of the things they do is they bind to several receptors simultaneously because they've got the same repeating subunits. And they cause those receptors to actually pull together. That particular phenomena is called capping. And then the antigen is pulled inside along with the receptors by endocytosis. And that causes an activation of those B cells to become plasma cells. And those plasma cells begin producing antibody. Anybody notice what's wrong with this illustration? This illustration has a mistake. Well, actually, once you differentiate into plasma cells, that's something I haven't told you yet, is that the cells stop producing receptors. And instead, they begin producing antibody. So that part's okay. What class of antibody isn't that being shown there? Could that be an IgG? The way it's shown right there? Valence of 2. Could it be an IgE? Could it be an IgA? That could be the serum form. But that can't be IgM the way they've shown it there. Remember IgM, it's 5 of those stuck together? And what do plasma cells that have been stimulated by T-independent antigens produce exclusively? Only produce IgM. Because they don't go through the class switching. And they can't produce other kinds of antibodies except IgM. So the illustrator of this should have shown these as IgM molecules, not IgG. I've used this slide for a couple of years now, and that was the first time I noticed that was this morning. Okay, so then those antibodies can help to clear the antigen. Okay, pretty simple, right? However, okay, go ahead.
Can you say that again? So they bind to like several receptors and they pull the receptors together and then what happens?
And that receptor and antigen complex goes through endocytosis. And then that is a stimulatory signal for them to start then secreting the antibody. Okay?
So they secrete it inside the cell and then export it?
Mm-hmm. Yep. Guys, remember that I said that one of the things that happens when these plasma cells differentiate is they just become filled with endoplasmic reticulum and they start cranking out antibody. Okay, I'm not going to show you this one in detail for now, but what this is showing you is kind of the T-dependent antigen pathway and the T-independent antigen pathway. Over here is what the T-independent antigen pathway is illustrating right here. In their particular illustration, this is happening in the spleen. The T-dependent antigen process that we talked about will be happening in the lymph nodes. Okay, so I'm not going to jump into that one right now. Okay, T-dependent antigens. They require the involvement of T helper cells. What's so important about the T helper cell? It's going to produce some cytokines, okay, like interleukin 4. And they're going to be necessary for the development of the B lymphocyte. Here we need 2 signals for B cell activation. One is binding the antigen. Okay? That's the only signal that this cell got. But because that antigen was T-independent, that was enough of a signal to produce modest antibody production. One class only. Here, the second signal, cytokines produced by the T helper cell, gives you a more robust response. And particularly, it gives you the ability to produce other classes of antibody and to produce memory. That's really important here, is that the T-dependent antigens cause a memory response. T-independent antigens don't. Okay. So what have we got here? Here we've got some T cells that have developed receptors. They're educated, but they're inactive. They haven't been stimulated yet. Here we've got an antigen-presenting cell. And when it finds the T helper inactive cell that it matches up with, here's all the receptors that are interacting. And here you can see the CD4 interacting with the MHC2. Remember, these are MHC2 restricted. So this has got to show the antigen with an MHC2 receptor. That gets things going. Okay? So these T helper not cells now start differentiating into T helper 2 cells. Those T helper 2 cells start producing an interleukin 4. And that interleukin is going to have an effect on the B cell eventually, but it has an effect on its own self. When they start producing interleukin, they start producing some other receptors which are necessary for the process to occur, but are kind of beyond the scope of what we're going to talk about. So we're not going to get into this particular kind of detail. But this cell now is activated. What's it going to do? It's going to bind to a B lymphocyte, which has already activated a T-dependent, or already bound, a T-dependent antigen. But that binding of the T-dependent antigen in and of itself is not sufficient to activate these cells into activity. They need another signal. What is that signal provided by? This activated T helper cell binding to this antigen that this cell is bound, the B lymphocyte, and now the interleukin 4 that's produced by this cell is affecting the B lymphocyte. And it causes to start growing and differentiating. And it forms a clone of cells, but because it got this cytokine signal, it's now able to produce plasma cells and memory cells. And so we get the full-blown immune response, and we get the memory response. So you see the difference. Over here, in the previous example of T-independent antigens, binding the antigen alone was sufficient to cause antibody production. In this particular case, with the T-dependent antigen, we've got to have the activation of the T helper cell first, and then that's got to bind to the B lymphocyte before the necessary cytokine, interleukin 4, causes the full response to develop. And we start getting the memory response and the plasma cell secretion of antibody. So what are you supposed to make of all this? Well, one, that it's complicated. Okay? And that there's a number of cellular interactions that have to take place. What's one thing that always has to happen? The immune cells, whether they're B or T, have to be exposed to their partner antigen, whatever one they have the receptor for. And remember, they randomly develop that receptor during the education process. Sometimes just being shown that antigen is sufficient to stimulate you into action. And that is the case of a T-dependent antigen stimulating a B cell. T-independent antigen stimulating a B cell. But usually you've got to interact with other cells, which then produce cytokines, to really get the full-blown response. So if you want to kill your own infected cells, it's not enough to just be showing an antigen. A T helper cell has to be showing that antigen, and that has to be activated. And then that has to produce cytokines, which eventually activate the T cytotoxic cell. Do you see why this one-step activation could be dangerous? Getting signal, a single signal that causes your own cells to start dying could really be risky. It would be like if one person only had to respond to a nuclear launch code and could push the button and destruction would occur. That's not the way nuclear launch codes occur. You've got to have verification, you've got to have multiple people involved to make sure that steps don't occur that would result in a mistake. That's why typically for activation of cells, you need other cells to be activated first. So we get this verification process. And then only if you get the multiple verifications is the launch code initiated that results in destruction of your own cells or production of antibody. This is one that I would encourage you to look at. The print's so small right here that I'm not really going to try to go through with this for you right now. But this kind of puts together the B cell response occurring to T-independent antigens and occurring to T-dependent antigens. Again, there are reference to some receptors and some interleukins on this slide that I'm not going to hold you responsible for. Basically, this is the information that you have to need to know in terms of being able to answer questions about this particular type of response. And then this is just another representation from another source showing the same type of thing. Down here is the B cell response. Down here is the T cytotoxic response. One of the real keys to this process is this step right here. When these T helper NOT cells are activated, if they go to T helper 1 cells, that's going to then favor the particular process of activation of T cytotoxic cells. If they differentiate into T helper 2 cells, that's going to lead to the pathway where they lead to activation of antibody production. Okay? So what determines whether this cell being activated goes this direction or this direction? Well, we don't know for all practical purposes, but there's some things that appear to be important. One is, what is the antigen presenting cell? Is it a dendritic cell or is it a macrophage? Dendritic cells tend to favor one pathway, macrophages tend to favor another. Where does the exposure to the antigen first take place with the antigen presenting cell? Is it in the tissues? Is it in the lymph nodes? That seems to influence the divergence of pathway. Because this activation is also controlled by various cytokines and various other molecules that are produced by the complement cascade or by inflammation. So this is really kind of a mess in terms of trying to make hard and fast rules about under this circumstance this happens, under this circumstance this happens. It's all controlled by kind of cytokine levels. And the cytokine levels are being determined by so many different factors. Whether there's an inflammatory response going on, whether complement is being fixed, whether mast cells are degranulated. So it really gets messy, and it's really difficult to kind of have linear thinking when you're trying to understand these particular processes. There's a lot of feedback, both positive and negative, and a lot of multiple interactions. So this is kind of the bare bones outline of what goes on, but it really is much more complicated than it's being illustrated here, and I'm sure right now your minds are going. Exposure. There are a couple of other diagrams I've got for you here that are just kind of flow diagrams which were very pared down. These are kind of things that can help you to kind of get a sense of the overall interactions that take place. And then this is one that kind of puts everything together for the first, second, and third line of immunity. Okay, remember for this exam, for the final exam, we only talked about part of the second line of defense, mainly complement, interferon, and inflammation, and then we talked in detail about the third line of defense. A couple of interesting things have emerged about the immune system that I think are worth mentioning kind of at the end of this. A lot of you are probably going to get sick within the next couple of weeks. If you're like me, you've been sick for the last month. Most of you, fortunately, will probably make it through your finals okay, but then oftentimes succumb soon thereafter. Anybody know why?
Has to do with like the cortisol hormone or something.
Yeah, stress. Okay, under final exam stress and stuff like that, your body responds to start producing various corticosteroids, and steroids generally depress immune system function. Okay, they also depress inflammation, and so one of the things you can get if you suffer from chronic inflammation is injections of steroids. What's that going to do? That's going to depress your immune response. What's another time when medically it might be important to suppress an immune response intentionally? Allergic reaction, okay.
Post-surgery.
Post-surgery, and particularly post-transplantation surgery, right? Because if there isn't a great match between the cell surface molecules of the donor and the recipient, then you can mount an immune response. So injections of steroids can be given to post-surgical transplant patients, but what's the downside of that? depression of your immune response. And so oftentimes those people will have to be in isolation. And the idea is during the isolation process, your body kind of learns to accept that particular tissue, and then you wean off the steroids, and then you start developing your normal, robust immune response. Okay? What they're doing right now is they're trying to develop specific drugs which are targeting T cytotoxic cells, because those are the ones that are causing the tissue rejection. And they're developing monoclonal antibodies, which are kind of like guided antibody missiles, which can deliver drugs specifically to the T cytotoxic cells and inhibit those temporarily, leaving your T helper cells alone, meaning you can still get this activation of your humoral immune response, even though you're inhibiting the T cytotoxic cells. So that's a very exciting field of study for transplantation rejection. Many of you have probably read that a lot of people believe that there is a connection between your ability to recover disease and your state of mind. And you might have even heard stories about people who have miraculous recoveries from cancers, for example, that doctors have said, you have a death sentence. And then all of a sudden, in a few months, the cancers are gone. Okay? This idea of relationship between state of mind, the nervous system, and the immune system is now gaining some real scientific traction. And specifically, what they're finding is that nerve cells can actually produce interleukins, like interleukin 2, and that they can have an effect on immune cells. Immune cells can produce compounds that affect nerve cells. And so even though these are two functionally different systems, they crosstalk with one another through the production of these interleukin molecules. And so there's a whole new science that's developed within the last decade called neuroimmunology, which really looks at the effect of the nervous system and the immune system on one another. The development of the immune system doesn't happen overnight. When you are first born, you have a very lousy immune system. It takes a while to develop. And part of the developmental process is exposure to all those foreign antigens in the world. We talked about the depression of the immune system that occurred when animals are raised artificially in germ-free environments, and they have a pretty lousy immune system. Some people argue that some of the diseases that are more common in your generation, like asthma, are the result of the fact that you're not immunologically as challenged as previous generations were. However, once you get up into puberty and through adulthood, your immune system is very robust, but then as you start to age, your immune system will decline again. And so I am in the declining feature of my immune system right now. And that's one of the reasons why there are recommendations for pneumonia vaccines, for example, once you reach a certain age, that are not necessary when you're young and healthy, because that immune system goes through a peak and valley, and right now you're really up at the very top of your immune system, but it will slowly start declining. It's a kind of a self-limiting process. So a lot of exciting things are happening in immunology. One of the exciting things about immunology is that antibodies that are produced by your body in response to infection can be used in various medical technology tests to diagnose the presence of that infectious agent. So that's the last thing we're going to talk about starting tomorrow, is the application of use of antibodies for diagnostic testing. So we'll talk about what some of the testing methods are and how antibody-antigen reactions are very important in those particular tests. So don't let all the detail of this kind of blow you away. Really use this sheet right here as kind of your guidance. And particularly the diagrams, I think, that are ones you should concentrate on are this one right here. That's figure 16 point, what's that, 18? Or is that 16?
Yeah, 18.
Okay. and this one right here, 16.14. Okay, the other ones are kind of reinforcing what those are doing. These in particular kind of put together things a little bit more visually. This is the same diagram right here. And then this one is just kind of another way of illustrating that information. So superimposing that on those two diagrams is something you should pay some attention to for this particular information. But don't get overwhelmed by this particular class lecture. Because remember, there's a lot of other stuff we've talked about that's going to be fair game on the exam too. So if you're struggling with this a little bit, this is a finite amount of points. And what I'm trying to do right here is kind of boil down for you what some of the really important stuff is that would help you to answer most of the questions to deal with this information. Okay? We get a 5-minute break. Yes?
Like any other foreign substance. So is that why some people are kind of immune, like they can handle a lot of snake bites and other people can't?
They've been previously exposed and have memory.
So you can basically immune yourself or immunize yourself from be immune to snake venom by just injecting small amounts of it.
That's the theory, yep. Yep.
We'll see you in an hour.
I figure out.
Ah, thank you.